Professor José Luis Mascareñas completed his chemistry degree in 1984, his PhD in 1988, with Professors Antonio Mourinho and Luis Castedo as supervisors. He was a postdoctoral fellow at Stanford with Professor Paul Wender and became a permanent professor in 1993 and full professor in 2005. He is at the University of Santiago and has been visiting a scholar at Harvard 2 times and at the University of Cambridge in 2009. In 2009, he received the Organic Chemistry Award of the Spanish Royal Society of Chemistry, and in 2010 became the first president of the Spanish group of chemical biology. His current research interests are split between a synthesis program focused on the development of metal-catalyzed processes and a chemical biology program aimed at designing new protein and nucleic acid recognition and sensing taxes. Mascareñas, it's your turn. Thank you.
Thank you. Can you hear me? Is working?
Okay.
No.
Yeah.
Now, can you hear me now? Okay. Good morning, everybody. First of all, of course, I want to thank Maria Jose for the invitation and for organizing this wonderful meeting. I have to say that we don't work exactly in nanomedicine, although we do work with proteins, nucleic acids, that in some way can be considered as nanomolecules, nanobiomolecules. When I gave the title, I was not sure about the format of the meeting, so it's a quite general title. But I will mainly talk about the work of my group in the area of DNA binding and DNA recognition. Well, I don't have to say that even non-scientists know that DNA is one of the more important molecules of life, if not is the most important molecule of life. And this is so because DNA is the main component of genetic material. Indeed, DNA determines the development, the functioning, and of course, the survival of the cells. And therefore, it has been a classical target for many drugs, particularly in the area of cancer. Indeed, many of the currently available drugs for treating cancer, anti-cancer chemotherapy, are still targeting DNA. This is the case of cis-platinum and derivatives that is widely used for the treatment of different types of cancer, particularly testicular cancer. It is believed that the way of working of cis-platinum involves the dialkylation of 2 guanines in neighbor positions of the DNA. This dialkylation promotes a folding of the DNA, and it looks like this folding induces the cell death, in general by apoptosis. Other DNA binding drugs that are also used for the treatment of leukemias, like anthracyclines, are interacting to DNA non-covalently by intercalation between the base pairs. There are many other interesting DNA binders. For instance, this type of DNA binder is called distamycin, and as you can see, it's a very nice molecule that binds DNA by inserting in the minor groove of a sequence rich in adenines or thymines. It's like, in some way, the cheese in a sandwich. So in this context, we were attracted by propamidine. Propamidine is like distamycin in the sense that also inserts in the minor groove of AT-rich sequences. However, if you pay attention to the structure, you can see that the propamidine is much simpler than distamycin A. Therefore, we thought that propamidine would offer us a lot of options to do easy manipulation, chemical manipulation. So we started to play with propamidine. The first thing that caught our attention is the presence of this chromophore. This is a conjugated chromophore. And we considered that perhaps this conjugated chromophore might allow to monitor the interaction to DNA by using fluorescence spectroscopy. As many of you know, in order to do fluorescence spectroscopy, what you need to do is to irradiate, excite the molecule. But unfortunately, the absorption wavelength of this molecule overlaps with that of the DNA. And this is a problem because it does not allow you to excite the molecule at this wavelength because you are exciting the DNA. But this can be easily solved. You just change the oxygen by a nitrogen. And in this case, now the absorption wavelength shifts to the right, and now you can excite this molecule without touching the DNA. The molecule can be easily made in 3 steps from commercial products, and we call it ASA-propamidine because it's an ASA version of propamidine. And as expected, the fluorescence of the molecule in the absence of the DNA is not high. Indeed, it's quite low. But gratifyingly, when we add increasing amounts of a double-stranded DNA that contains a sequence rich in adenines and in thymines, we observe an important increase in the emission of light. The data could be fitted to a 1-1 equation, and we got a dissociation constant of 89 nanomolar. That is not bad for such a simple molecule. But the better thing is that the compound interacts to DNA in a highly selective fashion. If you don't have the AT-rich binding site, the increase in fluorescence is much weaker. Indeed, very easily, we can test the selectivity of the interaction with different DNAs. And as you can see in the figure, if you have 5 ATs or 4, the interaction is good, but as soon as you decrease the number of A or Ts, there is a drastic decrease in the affinity. More recently, we have found that we can make synthetically another family of so-called bisbenzamidines in a single step from commercial products. So if you buy these 2 aldrich, it's an aminobenzamidine, and this dialdehyde that is also commercially available, and you mix both molecules in the presence of a reducing agent, you get in one step a quite good yield of this bisbenzamidine. And this bisbenzamidine is also able to bind DNA with quite good affinity, low micromolar affinities, and again, the selectivity is good. So the molecule is able to bind DNAs containing AT-rich sites. Very recently, in collaboration with Professor Jesus Jimenez Barbero in Madrid, we got an NMR structure of our molecule with a double-stranded DNA that contains the AT-rich site, and as expected, the molecule inserts in the minor groove of this DNA, of course, in the sequence AATT. Well, this is another view of the molecule in which you can identify the different aspects of the structure. This is the phenyl ring, this is the amidinium, and the amidinium is sitting here, touching the floor of the DNA group, and this amidinium is critical for the interaction to the DNA. Well, we have a very easy synthetic method, one step from commercial products. If we use other type of dialdehydes, we can make many other derivatives. For instance, we can very easily make this derivative that contains an SI group. I'm sure that at least the chemists in the audience know that asides are excellent groups for bioconjugation reactions. So we could introduce here different functionalities by doing a standard Lick cycle addition chemistry. Considering the binding mode of our molecule to the DNA, this aside should be here. Perhaps you can see it better in the model. So the aside is just protruding out of the group of the DNA. Therefore, it provides an excellent linking point to introduce different functionalities just in the surface of a specific AT-rich sequences. We have linked different groups to this aside. I will show just one. In particular, we conjugated our aside with this alkyne that contains a coumarin fluorophore. Therefore, we can very easily make this conjugate in which we have the DNA binder and we have the coumarin fluorophore. This molecule is quite interesting because while the compound that contains the aside is not highly fluorescent, if we excite with light the conjugate, what we observe is a very intense fluorescence in the blue region of the spectrum. What is happening here? Well, what happened is that we are exciting the DNA binder, but there is an energy transfer from this chromophore to the coumarin, and is the coumarin the one that is emitting the light. This is interesting because it allows you to detect DNAs containing this type of sequences, even with the eye. With the naked eye, you can detect nanogram amounts of these DNAs. If you don't have the right sequence, the emission of light is much weaker. So what we have here is some kind of optical sensor of AT-rich sequence of double-stranded DNA. So you don't need to separate the strands of the DNA to detect the sequence of the DNA. I have to say that these molecules are also interesting from the biological point of view. Indeed, in collaboration with Dr. Rivas in Madrid, they have found that some of these molecules show very interesting anti-parasite properties. In particular, this compound with the SI group shows good anti-leishmania activities, and this other that contains the propyl-ammonium tether shows nanomolar affinities against malaria. We are further studying the pharmacological possibilities of these compounds. Okay, at this point, we thought, well, we have a very easy access to this type of molecules. Can we introduce some molecular trick that allows to cancel the DNA binding of these molecules? You might ask, why do you want to cancel the DNA binding? Everybody wants to get binding, not to cancel binding. Yeah, okay, we want to cancel the binding, but only in a temporary manner, so that we can recover the binding when we want. So the question is, how can we cancel the binding of these molecules to DNA? Given that for the interaction to the DNA, this group is critical, the amidinium group is this group here, perhaps one way to cancel the interaction could consist of introducing some blocking, some caging group in the amidiniums. If this caging group is photolabile, maybe with light we can remove it and we can recover the binding. And therefore Matteo was able to prepare this diprotected derivative with the caging groups, and indeed with light, with UV light, this group is photolabile, so with UV light you regenerate the parent azapentamidine. As expected, the compound that is diprotected is not capable of binding to the DNA. The fluorescence emission is weak. However, if you irradiate light on this compound, then you are able to recover the emission of light. So we have a light switchable minor groove DNA binder. We have even coupled this binding, this switchable binding to our biological activity. In particular, we tested the ability of our compounds to inhibit the growth of Candida albicans. You know Candida albicans. It's this fungus that originates many opportunistic infections in humans. We were surprised to find that azapentamidine is very active against the growth of the fungus, almost even more than the commercial antibiotic clotrimazole. But remarkably, the compound with the caging, with the protecting groups, is inactive. The fungus can grow. However, if you now take this compound and you shine light on it, we recover the antifungal activity. So we have a light activatable antifungal agent. I'm not sure yet if this is going to be useful or not, but at least it's funny. Okay. The next thing that we thought is in extending these caging strategies to other amino groups, to other DNA binders. And in particular, we consider ethidium bromide. I'm sure that many of you know ethidium bromide. Ethidium bromide is a cytotoxic molecule that also binds to DNA by intercalation. But the more interesting characteristic of ethidium bromide is that when intercalates DNA, is capable of emitting a characteristic red fluorescence light, and therefore is used in many laboratories to detect, to stain double-stranded DNAs. Matteo also showed that if you protect these amino groups of ethidium bromide with these caging photoligand groups, the molecule becomes inactive. And again, if you shine light on the mixture, you will recover the palin ethidium bromide, you will recover the fluorescence. Well, what happens if you treat cells, in this case, Vero cells, with ethidium bromide? When you treat cells with ethidium bromide, you go to the microscope, to the fluorescent microscope, you see nothing. After, you could see staining of the nuclei inside the cell, but you see nothing. What happens? Well, this molecule is very polar and is not capable of entering the cell or crossing cell membranes. Of course, and then it cannot reach the nuclei, and therefore we don't see staining of the DNA. Of course, our derivative that contains the protective group is also not able to stain the DNA because this molecule is not even able to bind to DNA. However, if you take these cells that have been treated with our derivative and shine light on them, after 10 minutes, you can see a clear emission of red fluorescence from the inside internal DNA and RNA. So what is happening? What happened is that this molecule, in contrast to the parent ethidium bromide, because of the presence of these protecting groups that are hydrophobic, are able to cross the cell membranes, and they remain inside the cell until we wake it up with light. The strategy can also be extended to other DNA binders. This is very famous also, DAPI. It's used to stain DNA nucleic acids in cells. If you protect DAPI, the amidiniums, the molecule becomes inactive. This is inactive, this is active. This can be easily observed in cells. If you treat also cells with this DAPI derivative, at the beginning you cannot see the nucleus, but with time, with irradiation time, you can see a clear emission of blue light coming from the cell nucleus. Yes, Matteo is going to inject a dose of our DAPI. He's going to inject a dose of this compound that is inactive to the mouse, and then he took the mouse and irradiated just one of the eyes of the mouse. Now, unfortunately, he had to sacrifice the animal, extract the eye, and check the retina, and he observed that only in the irradiated eye we can see a staining of the retina. So this experiment is saying us that we can activate the molecule in living animals and using external irradiation sources. So we activate the drug in animals. Okay, next step, we thought that it could be interesting to extend this type of activation strategy, of conditional activation strategy, to more complex systems. In particular, we thought on transcription factors. Transcription factors, as Professor Wong commented, are very important DNA binding proteins that, upon binding to DNA, they are able to promote the action of the polymerase, the RNA polymerase, that now is capable of promoting the production of a protein. It is well known that in many cases, these transcription factors are inactive in the cytoplasm or in the nucleus and become active and capable of DNA binding only after receiving an external signal, usually a ligand. Well, this interaction of transcription factors with the DNA takes place using different structural motifs. And one of the more simple and efficient motifs is that used by the so-called BCIF type of proteins. These proteins interact to DNA by making a helicoidal dimer. We can see that there are two regions in the protein. This is the dimerization region. It's called leucine zipper. And we have a DNA binding region that is called basic region. As you can see here, the basic region is inserting in the major groove of the DNA. This is the major groove, and this is the minor groove. It has been shown that it is possible to replace this leucine zipper by a covalent dimer without significantly compromising the DNA recognition of the system. If you look at this binding from other point of view, this is the front view, this is the DNA, and this is our dimer, the dimer. We can see clearly that there are several amino acids in the protein that are key for the interaction to the DNA. These amino acids are basic amino acids, therefore this region is called basic region. Basic amino acids are positively charged amino acids. So we then wondered whether we could cancel this interaction by quenching this positively charged amino acid. And the best way to quenching a positively charged amino acid could be by introducing a negatively charged tether. For instance, a glutamic acid tether. If you introduce a glutamic acid tether, you are canceling the positively charged guanidine of the adenines. If the linker between the tether and the basic region is photolabile, you might be able to recover the interaction by irradiation with light. Well, we have been working in this project for several years, and finally Adrian was able to prepare a model that works quite well. This is the model. So here we have the dimer between the two basic regions. These are the basic region, so the amino acid, the peptide that binds to DNA. This is the photolabile linker, and these are the glutamic acid chains. Then we tested the system by electrophoresis assays with a double-stranded DNA that contains the binding region. And as you can see, if we don't have the tethers, the dimer is able to bind DNA quite strongly. This is the complex between the peptide, the dipeptide, and the DNA. If we have 4 glutamic acids in each of the tethers, we still see the formation of the complex. However, if we have 8 glutamic acids in each of the tether, we completely destroy the DNA binding. But the good thing is that if you take now these samples and excite them with light, with UV light, you are able to fully recover the DNA binding of the molecule. So what we have here is some kind of model that mimics the natural system. In the natural system, the activation of the transcription factors usually takes is because of the action of a ligand. In our system, the external stimulus that is activating the DNA binding is UV light. So we hope that this strategy can be extended in the future. We are working on that to control or to suppress the expression of specific genes and cells. Finally, let me comment that we are also working in the development of biosensors of different types of molecules. For instance, of transcription factors. This will be important because it's known that many transcription factors are wrongly expressed in different types of diseases. So it would be nice to have a sensor, a biosensor of specific transcription factors. For instance, of Jun. Jun is a transcription factor that is considered as an oncoprotein, and it looks like it is overexpressed in different types of cancers. So it would be nice to have a good sensor for Jun. The mechanism of action of Jun involves the formation of a DNA complex with its partnered force. It's called an AP1 complex. So now the question is, okay, how can we make a sensor that is specific for Jun? Well, there is a very interesting property of this type of dimers, is that these proteins, when they are monomeric, they are not helical. They have no structure and become helical only after the formation of the helix. So this is an interesting property. With something like that. It's a folding process. So we thought perhaps we could take advantage of this folding process to develop our sensor. And we indeed considered that one potential sensor of Jun will be based on a phos type of receptor that contains two fluorophores that are relatively far away, but upon formation of the complex with Jun, these fluorophores will be closer and we will see the emission of light. And this should only happen if we have Jun in the medium. Well, I won't go into details, but we use as a fluorophores tryptophan, that is a natural amino acid, as donor, and as a sector we use a lanthanide complex because they are very good emitters in the visible. Well, this is where we introduce the groups, the tryptophan. They have to be in the outer side of the coiled coil in order not to interfere with the dimerization. Well, this is our sensor. It's a synthetic one. We have the tryptophan here. We have the lanthanide. It's a terbium complex in this position. And, well, the first result was not as good as we wanted because we wanted no signal by the sensor by itself. It has some signal, some emission. But anyway, we found later that upon addition of the oncoprotein Jun, we observe a clear increase in the emission of light. Something that does not happen if you have other proteins like phos. So the sensor, well, this is another type of experiment, circular directories, that also test that the system is working. So we have a sensor of Jun. It's not extremely sensitive, but it's highly specific. And we are working in trying to optimize the sensitivity of the system. And more recently, we are also working in extending this concept of folding sensing to other biopolymers, in particular to RNA. So I hope I have shown you how, using the basic tools of synthesis, I am a synthetic chemist, we can construct what I call functional artificial biomolecules that are of interest on the frontier in chemistry and biology, perhaps in the future also in medicine, in therapy, diagnosis, and perhaps theranostics also. Let's see. So, of course, this work has been possible thanks to my co-workers, Eugenio Vázquez, that was my first PhD in this field, and is now a professor and is supervising the group with me. Olaya is doing a postdoc in Berlin. Elena has joined just the group of Professor Stubb in Chicago. Adrian defended the thesis 3 weeks ago. And Matteo is doing the PhD with me. Right now he's doing a short stay in MIT. I also want to thank my collaborators, particularly Professor Martinez Costas in CiQUS, because he's helping us with all the experiments with cells and the molecular biology experiments and the funding of the Ministry and the Junta. And thank you for your attention.
Thank you, Professor Mascareñas, for the talk. Any questions to Professor Mascareñas?
Okay, so that was a very nice talk. So with the ethidium bromide, I assume you can use that principle for other drugs too, or sensors. So is it, I'm assuming that the modification will go into any cell type, is that correct? Or
into any cell type, to me.
Like it's basically crossing the cell membrane,
Yeah.
is that correct? Have you, I guess that would be okay, because then you could maybe shine the light specifically only on particular area, but have you thought about modifications where it would only go in cell specific, or is that very difficult?
If you tell me about that, I will be happy to test that, because this is something that will be interesting, but you know, we are not sure of about what will be the most interesting aspect to check, so this is something that could be useful for us.
Of the group,
Yeah. Yeah, but we are exciting the bisbenzamide, 320 nanometers. It almost doesn't touch the coumarin. Okay.
To activate the molecules in vivo, like in the mice, you have caused any damage in the DNA, because DNA is an ultraviolet sensitive molecule. In fact, many cancers comes from some exposition to ultraviolet. So maybe you are damaging.
This is a good question. It's true that DNA is sensitive to UV, but in our cases, the idea is trying to go to the longer wavelengths. So we try to extend the molecules of the chromophore so that we can irradiate the cells with longer wavelengths, so that we can avoid that. So this is a proof of principle, proof of concept, but if this works, of course, it will be very nice to go to longer wavelengths. One way to go to that is using the 2-photon spectroscopy. With the 2 photons, you can irradiate a longer wavelength without damaging the DNA. So, but the first thing is to prove the concept.
So, you, I don't remember the name of the molecule you have synthesized, but the one that sticks
There's a bisbenzamide, yeah.
Okay, so when this molecule sticks to another DNA molecule, not only one, right?
Yeah.
Okay, the final result is a nanostructure.
Well,
Have you ever measured the size of this the
not really, because what we do, everything is in solution, so we don't not sure what happens there. But as you can see in the NMR, we are able to solve in solution the structure of the DNA complexes with our molecule. But there is one thing that we are doing now in collaboration with other people, is we are using our molecules to introduce nanoparticles, so that you can codify with the DNA the introduction of nanoparticles in different parts of the DNA. Because we have code-reading DNA molecules, so you can take any nanoparticle, anything you want. You can take it and drive it to any position of the DNA. Well, not to any position, in this case to 80 rich sequences. But the idea is modulating our molecules so that at the end of the day we can have nanoparticles that go into any part of the DNA.
Is the next question would be like this kind of association? Do you think it would help the internalization of DNA to the cells? I mean, like
could be.
binding molecule could be a carrier
Could be.
for DNA because you're neutralizing the charge of DNA.
Yeah. You can imagine, for instance, that you could have a big DNA and we could have two of these molecules linked, so that in this way you induce the folding of the DNA, because these molecules are going to bind to different sites. So to induce the folding, you will have a nanostructure of DNA, and what I don't know if this nanostructure is going to cross the cell membranes better or not, but you could use the molecules to modify the DNA nanostructure. Yeah, sure, I'm sure that.
Could be.
Okay.
Oh, wait,
Just time. Sorry.
Just to follow up on that, so I then showed the switch one way. Can you go the so if you're then binding, can you release then? I'm just wondering, yeah.
We have used it in the past, isobenzenes. Isobenzenes allow you to switch one way and the other way. In this case, no, but you can do it.
Okay, thank you very much, Dr. Mascareñas. Let's go with the next speaker, which is Professor Eduardo Fernández Mejía.